Okay, guys, we're talking photosynthesis today. This is going to be a long PowerPoint. I'm going to try my best to zip through it as fast as I can. You might have to pause it along the way to get some stuff down. I might even have to do it in two videos. We'll see. Okay, so the whole purpose of photosynthesis is to take light energy and inorganic compounds like carbon dioxide and convert them into usable glucose for the plant. So carbon dioxide plus water yields glucose and oxygen. And we're gonna kind of go through where all these things are coming from, how they're getting used, how they're getting made. Okay, so um, photosynthesis and cellular respiration are considered complementary processes because the products of one are the reactants for the other. So whatever one makes, the other one uses. They're not opposite because they don't do it in the same steps. So the steps are very different on how they produce those molecules. So they're not opposite, but they're complementary. So autotrophs make their own energy. So they can't go to McDonald's, grab a cheeseburger. They have to make their own energy. And to do that, they utilize light energy, they capture it, and then they create their own form of organic substances from inorganic substances from the atmosphere. So they're using sunlight to create their own energy. That's an autotroph. All right, if you take a look at a cross section of a leaf. And remember, photosynthesis happens in the leaves. So here's a cross section of the leaves. There's a couple important parts of the leaf. So this part right here is the stomata. So the stomata, they have these cells on both sides. So they're kind of like guard cells. And they can open and they can close depending when they want gas exchange to occur. So these are like the little pores in the leaves. They can open up and allow carbon dioxide in and oxygen out. At the same time, when they open up, they'll transpire too. So they'll let a little bit of water out. If it's a really hot day, they'll end up losing a lot of water through the stomata. Remember, that's called transpiration. Okay. And inside of the mesophyll here, there's these mesophyll cells. Here's an example of one of those cells. And in those cells, they have those little green dots. These little green dots are actually the chloroplast. So this entire thing is considered a chloroplast. And this is where photosynthesis takes place. So inside of the chloroplast, we have a few important components that I kind of asked you to go over already. So we have the outer membrane and the inner membrane. So this is a double membrane organelle. Inside is the stroma. So it's the fluid inside the stroma. It's kind of like the cytosol of the chloroplast. And these guys here, they kind of look like stacks of coins. Each individual coin is called a thylakoid. And if you stack them up together, each one is called a grana. Okay, so you can see it a little bit better on the next slide that I show. So if you want to fill in your diagram, so each one of these stacks is a thylakoid, or sorry, each coin is a thylakoid, each stack is called a granum. We got the outer membrane, inner membrane, and the stroma. And if you look at this as an individual thylakoid, each thylakoid has its own membrane, which is important. That's where light photosynthesis is going to take place. And inside each thylakoid is a solution called the lumen. Okay? So outside of the thylakoid is the stroma. Inside the thylakoid, that liquid is called the lumen. Okay, when it comes to the nature of light, I asked you guys to do that online lab to get a better understanding of what color light plants like best. And green plants actually don't like green light. So you'll notice that they don't grow as big in the presence of green light. Reason being, it reflects green light. What you see is light reflecting off of something. So if you look down, you had a red shirt on, that portion of the wavelength hits your shirt, reflects back in your eye, and you see it as red. It doesn't absorb it. It absorbs all the other colors. Um, so remember, light is a form of energy with different wavelengths. Red is the longer of the visible spectrum. Violet is the shorter of the visible spectrum. The shorter the wavelength, the greater the energy for each photon, and certain wavelengths are detected by the human eye. We can only detect this portion in here, this visible portion. There are all these different wavelengths going on around us right now. There's radio waves that are flying by our head. We just can't see them. We would need an antenna in a car to pick these up. We can only visibly see this portion of the spectrum. And it's the visible light or that size wavelengths that are going to drive photosynthesis. So let's go here. So if you guys remember, here's in each thylakoid has its own membrane. And inside that membrane are where we're going to find these pigments. Okay? So this is what a pigment looks like. So at the top we have a porphyrin ring with a central carbon atom. This is what absorbs the light. So this is actually on the outside. If you see these little green dots here, this is the porphyrin rings. They do the absorbing of the light. And then this phytotail, it's hydrophobic, so it anchors nicely into the thylakoid membrane, so it stays there. So this portion here anchors into the hydrophobic tails, and it kind of sits there, and we got the porphyrin ring up here ready to absorb light. Okay. So absorbing light pigments. Chlorophyll is green because it absorbs mainly blue and red wavelengths. It reflects green back. So most plants are green because they have chlorophyll in them. Carotenoids, sounds like carrot. Okay, they have carotenoid pigments that absorb mainly blue and green light, and they reflect orange and red back up. So if you look at the visible spectrum here, when you guys did the lab, you should have seen something like this. Different chlorophylls really absorb the red and the blue. So the violet and blue, they absorb a ton of that, and they absorb a really low amount of the kind of green yellow spectrum in here. So plants don't like the green light. Okay, so if you notice in the fall that the chlorophyll pigments actually break down and they leave the carotenoids there, so that's why leaves look yellow, they look orange, they look red in the fall, because that chlorophyll pigment break down. Okay, so there's two stages of photosynthesis. There's the photo stage and there's the synthesis stage. Today we're just talking about the photo stage. So photo means light. So these are light dependent reactions. They are going to create the energy that we need to make and carbon fix molecules to make glucose in the synthesis stage. So today we're just talking photo stage. So this is kind of what it looks like. So we're talking light reactions. In light reactions, we utilize the water. Remember our equation from photosynthesis? So we utilize this water, and in the light reactions, we're actually going to produce the O2. So we're using water, producing O2, and we're utilizing the sunlight. We're going to create ATP and NADPH for the Calvin cycle, or the synthesis cycle. This is where we're actually going to make sugars by fixing carbon. Okay, so let's get into that. So what is the main purpose of light reactions? Well, we're going to input water. We're going to grasp those photons of light, and we're going to output oxygen, ATP, and NADPH. So NADPH is like the exact same thing as NADH. It's just a high electron energy carrier, just given a different name in plants. So three parts to this. So photoexcitation. So we're going to absorb light. We're going to excite the electrons. Those electrons are going to go through an electron transport chain, where we're going to harvest the energy from those electrons. That electron transport chain is just like the electron transport chain we learned in cellular respiration. We're going to pump hydrogens into an electrochemical gradient. We're going to use that electrochemical gradient to phosphorylate ATP, or make more ATP. All right, I'm gonna whip through this really quick. When an atom absorbs energy from the sun, it can become excited. It can jump. It can jump to a higher state. We need to find a way to capture those electrons. If we don't capture them, well, they're going to come back to the ground state. They can be released as heat, or they can be released as a photon of light. If you have a fluorescent gene, unfortunately, we're not going to be able to do our glow-in-the-dark bacteria, but some living things have a fluorescent gene, which means they're able to give off their own light. Like an anglerfish or a firefly, they produce their own light. We were going to take this fluorescent gene, put it into a bacteria. That bacteria was actually going to glow in the dark. So they're doing a lot of things now where they're taking different genes and they're placing it into different organisms. So I bet by the time you guys are parents, I think it would actually be somewhat normal if the streets were lined with fluorescent trees instead of street lights to produce their own light. Just a guess, but they're working with it right now, where they have produced trees that give off their own light. They absorb it during the day, and they actually give it off at night. Let's just work through this. Okay, so a photosystem is just a bunch of pigments where a photon of light comes in, it gets excited, and it gets trapped by this electron transfer. So this is the primary electron acceptor here. So electrons jump till they get to a high state, and then they get captured. Okay. Once they get captured, they're able to move through an electron transport chain. I'm just ripping through these, okay? So there are two photosystems. There's photosystem 1, or P700, and this is activated by a photon of 700 nanometer wavelength. Then you got photosystem 2, or P680, and this one is activated by 680 nanometer photon. Okay. So you're going to hear me say the Z diagram, or the Z scheme. So photosystem 2 starts things off, and then photosystem 1's over here. Photosystem 2 is first because photosystem 1 was discovered before photosystem 2. So photosystem 1 is first. So in photosystem 1, we take this electron and we excite it. Right? We capture the photon of light and it excites these electrons. These electrons jump up and they find this electron carrier PQ. And it goes through an electron transport chain. And as they keep passing down, the electron loses more and more energy. So you kind of notice that it's falling down a little bit. And as it falls down, it actually drives the synthesis of ATP. Then it gets down here where it's lost quite a bit of energy. Photosystem 1 reabsorbs some more photons of light and excites that electron back up here to another energy carrier. This would be like ferredoxin. And then it goes down to NADP plus to get reduced into NADPH. This is the Z scheme. And if you notice, like maybe I can turn this. If I turn this picture this way, it kind of looks like a Z. Okay, so that's why it's called the Z scheme. Okay. So just an overview of what's going on here. We take the water inside the thylakoid space. That water is split using light. Okay? Then the electrons from the water are excited in photosystem 2. It's going to move to PQ. past the cytochrome complex, and when that electron passes by it, we actually pump hydrogen